Hey everybody. Playing around a little bit, trying to get the iPad set up so you can see it. My daughter is drawing behind us on it, and it seems to be taking a while to find it. And I'm not sure why. It's like it can see it, it just refuses to put it in. Haha, there it is. Now you should be able to see it in the background behind us. So she was interested in doing some drawing, and I said, well, I'm going to be doing some microscope streaming, so we're doing a collaborative stream. She's back here behind me in the chair. Drawing on her iPad, which is fine. Yeah, this is some material that I collected today. I went out with students all weekend long, and so 10 in the morning until basically 4 p.m. both days, Saturday and Sunday, we were driving back and forth between, well, I was driving back and forth between the university and the field and picking up students and bringing them out. And it's because my class is too big for everybody to use everything at once. So I also thought I would take a little time and record what's living in the samples, because probably by the time we actually get to look at them for the class, everything in the samples will probably be decaying. I'm not sure, but it definitely won't be, most of them won't be alive, because they're collecting plankton today and yesterday, but they the projects that they're working on are over the next 2 weeks, and so the samples are probably not going to be very healthy after today or tomorrow. I think most of the stuff will probably be reaching a conclusion in a day or two. Even in the refrigerator, they might not last very long, because some of the students had very concentrated material, and I tried to explain to them, you can't just have, a ton of stuff all in the same sample that won't have any oxygen. And I think various bits of that story actually made it through to what they were trying to understand. So I was like, it's alive today, it'll probably be dead within a day or two, even if you keep it refrigerated, because it's not a natural condition to be inside of a centrifuge tube. So so we can look around a little bit while my daughter is sort of behind us doing the drawing in the corner that you're seeing. I think she's working on some birds or something. I'm not sure what the algae is that's the main algae that's floating around in the water column here. But there are a bunch of zooplankters. There's this one. Oops, let's see. It's shaking just a little because I'm leaning on the desk. This one that you can see, which is a copepod, probably used to seeing them from Pacific Plankton Stream, on a different microscope, of course. I'm going to just reposition this one. You can see that it's still alive because its internal organs are still moving around. It's digesting stuff. Many copepods have a single red eye in the center of their forehead here, and then you can see it's digesting stuff here. These are these appendages that are coming off the front, sort of antennae, usually define groups of copepod by a little bit by their body size, and then the length of these antennae up here. And what you can't see is that they have sets of legs that are under the body in this part, and it usually allows them to move very, very quickly. And then these appendages back here on the back end are basically their sexy parts for the most part. And they will often find them basically carrying their eggs back here as well if it's a female and they had eggs recently. A lot of the things that are in these samples actually are carrying around a little egg because the lake was just filled with stuff, like all kinds of plankton. And it's about 10 degrees, which I think is right around where the zooplankton start hatching in the lake. So we're approaching the clear water phase, which is basically what happens when all the zooplankton hatch and they start eating all of the algae. You can see whatever this long skinny stuff is that they're not really consuming much of it. But there's also a bunch of rotifers in here. This is a rotifer that's swimming around in our field of view. Let me zoom back out a little bit. Let's see if I can keep it in the field of view for very long. Long enough to focus on it. There we go. It's just sort of swimming back and forth. So I've never seen these rotifers before, and I didn't know what it was at first. But you can tell that it's a rotifer because around the head there's a set of cilia that it's using to swim around with. So there's also these copepod nauplii. So the nauplius is the juvenile form of the copepod that we saw. They look a little bit like, I don't know, like a spider or something, but they are clearly they have the same body shape in this part of their body, the main part of their body, and then the little swimming legs that they have that stick off are, not obvious in adult copepod, and you don't see the long antenna coming off of them, but they still have the red eye in the center. If I ever watched the Clone Wars, rotifer remind you of a droid ship? I don't think I have. I don't think I've ever seen the Clone Wars. That's the animated version of Star Wars, I think. I haven't seen that. And then this little guy right here is a dinoflagellate. So we sometimes see these, and often Del Maximum has a bunch of these in his samples. So this little spaceship-looking thing right here is a dinoflagellate, a freshwater dinoflagellate. It has 3 sort of spines coming off the back end, and then a long curved front end. And I guess Ceratium is probably its old name. I don't know if it still has that name in freshwater systems. Part of the reason, as I mentioned, I wanted to analyze these or look through these is try to see what we could find in them today before things start to get bad. And we just end up with a bunch of debris in there. But this little star-shaped thing right here is a diatom colony. So this is Asterianella formosa, and it's a ring colony. They attach like this with their foot poles and then radiate out from the center. So Codon, thank you for following. This little thing that's right here, actually, this little fragment of a thing, this is Denobrian. It's a type of golden algae, but I think this one is dead. They also live in colonies. Hey Nid, how's it going? We're looking at some samples from a field trip that I took this weekend. It's a close by place, so it's near the university, but I was out in the field with students, basically. I found another rotifer right here. Can keep it in the field of view, especially with that on. It just sort of buzzed in and out, and I lost it already. Part of the issue is that they move around pretty quickly. So this is that long skinny rotifer that we saw. Let's see if I can get it all in the field of view. Stoned Boomer, hello. There goes one swimming by. And this is the This one I think is dead right here, but you'll see them go swimming by. They have this very long shape. But if you watch this part, you'll see that they have a ring of cilia up here around their head, and they're swimming through the water column by spinning it around. Hey, Dr. N, how's it going? Nice to see you. Here comes one. Here comes one into the field of view a little bit, and there's another rotifer right here, which is the one I was trying to get in the field of view before. There it is. It's just a little too quick for me. You can see there's another one that's swimming around right there. Those long skinny guys, the spaceship looking ones. In a lot of ways it's probably better if I just sit still and let them come to me and then try to focus it, because they're moving around quite a bit. Here's a rotifer with it's carrying its egg. You can see, I think you can see now the stop moving quite so much. The head has a sort of cilia that it's using to move around. Hello, Devil and Mrs. J. Welcome in. Welcome back. I've got a button for that. You could do me a favor and give Mrs. J a little follow. She does some crafting on Twitch. And some interesting pixel art sort of stuff that she does. Haven't seen you stream for a while, Mrs. J. Here's one of those rotifers. I'm trying to just keep them in the field of view, and they keep kind of swimming, but you can see there's an elliptical shape there attached under around the tail spine, and that's the egg that it's carrying around. And I think if I can keep this one in the field of view for a little bit, you'll be able to see the front end of it has cilia that it's spinning, which is the corona. The star-shaped thing again is the Asterionella formosa. Here's a different type of rotifer. If it's going to sit still like that, I'll be able to zoom in on it. Oops, not with that button, I won't. With this button, I will. There we go. And you can see the little corona on the front end of the organism that it's spinning, and it uses that to basically drag itself around. So it's moving those hairs pretty quickly, and you can see that, so they just basically wiggle them around very rapidly. Hope to get back into it soon. Okay, cool. Well, I mean, I'm not trying to put any pressure on you. I was just pointing out I hadn't seen it for a while. But you can see that this is a rotifer by the coronal end, the anterior end. Up here you can see the little hairs that are wiggling around. They can use those to draw food towards their mouth, which is in the center of that set of a ring of hairs. And then they can also use that to propel them through the water. One of the things that separates a lot of the rotifers from other organisms in ponds, aside from the copepods, most of the rotifers are actually pretty good at moving around pretty quickly. So for benthic organisms that we usually find in the benthos, those ones are pretty effective. These are planktic rotifers, and so they have a little different shape than the ones that we usually see, which look like little tiny vacuum cleaners with like spinning cilia coming off of both sides of their heads. And there's some more planktic rotifers in here. The ones that have these sort of long spines sticking off the front and the back they're all planktic rotifers, so they have a little bit different body plan. But it's cool to see some different things. We collected these with a plankton net, so from a boat, and one of the things that the students noticed when we were out there is that this sample had more of a yellow appearance, And the other sample that we collected from the same lake, but the other end of it had like a very green kind of color to it. There's a rotifer. Just looks like a set of little spines basically, but if you can see right at the front end of it, basically you can see little tiny hairs moving around in the same way. And there's a bunch of different types of those. At least two of the long skinny types are pretty common in the sample. I don't know what type they are. Haven't tried to figure them out. I'm hoping that the students actually spend a little bit of time trying to figure out what the dominant organisms in this system are. There's a nice clean view of a dinoflagellate. That one's, I think, probably not living anymore, but we can then zoom in on it, which is nice. So you can see they kind of look like a caltrop or a jack of some type. That was a rotifer that swam through. All dinoflagellates have a furrow, a little a depression in the outside of their skeleton. And their skeleton are made of a set of overlapping plates. So there's a plate here and a plate here. Each of these things are made up of a set of plates that are stacked on top of each other. So this is the outside of their theca. And then they have furrows. One runs along the length of it vertically usually and gives you an axis control in the Y direction, and one of them gives them another one usually goes around like a belt around the outside of them and gives them sort of X control. And then they keep their cilias and our flagella in the little furrows, one in each of the furrows, and it helps them basically focus how they swim. So they're usually very targeted, active swimmers, but they can't swim well enough to fight a current, so they usually just use it to basically stay in the light. This one I think is mostly planktonic and mostly photosynthesizes. Some dinoflagellates can actually switch their food sources between photosynthesizing and being heterotrophic and eating other organisms, but I don't think this one does any consuming organic debris or any other organisms. I think these ones are exclusively generating their own energy from sunlight. And this Copepodnopoleus has butted its way in, just doing a little photobomb of its own right there. How's it going with the drawing back there? Good. I have these little reminders that pop up as I stream that point out other people that you should check out. And this one is a science streamer that popped up, which is Jolkson. And Jolkson is a long time now. He's been working on presenting stuff from Microscope streams on Twitch. So he's a fellow Microscope streamer. And he doesn't talk a lot in his streams. He usually just puts the organisms up there and then moves around and has some nice music playing in the background. So pretty low-key, chill interaction. How are dinoflagella genomics these days? Dr. N, I don't actually study anything with genomics, so I don't really know anything. And I mostly study diatoms, not dinoflagellates, so I'm pretty poorly equipped to answer that question. I found another rotifer here. Ooh, let's see if I zoom out, you might be able to catch a little bit more of it. Oh, I think I lost it. That little rotifer was just making its way through the sample. Oh, here's a ciliate. I haven't seen many ciliates yet, so here you can see this one's very much like a vacuum cleaner head. It's just spinning. For the most part, I'm a paleoecologist, so I mostly study the skeletons of diatoms, and I still function as a limnologist pretty well, and a little bit of as a taxonomist, but I never do any of the genetic stuff. It's always the morphology and the skeleton that I focus on. largely because I work in samples that span back to, you know, 20 million years ago, where the genetic side of the story often doesn't help. Or I'm looking at ancient stuff that maybe they could get at with ancient DNA, but we're still sort of working on developing those techniques. So, and I have colleagues who specialize in that a little bit more than me. So, pretty underdeveloped answers for anything that deals with the actual genome stuff. Hey, Disillusioned, how's it going? Yeah, this one is a little bit like a little moon that's spinning around. Here you can see the cilia, the little tiny hairs that it's spinning around very rapidly around the outside of its body. They're not actually spinning around. They look like they are. Same with most organisms that have cilia. They just have an organized fashion of flicking them up and down such that it looks like it's rolling around the outside or spinning around the outside. But they don't do. They don't actually. It's not like a wheel that's spinning. It's just whirling around. Miles per hour. Hello. Are there any aquatic inverts in the water? Well, there's a lot of aquatic inverts in water. I don't know that I caught any in the plankton. Most of the time, the aquatic invertebrates are in the shallow water. But I also collected some material from the shallow water. So once I get a chance, and I've sort of documented all the little weirdos that we have floating around in the water, I will pull out some of the benthic stuff. And I've often found chironomids, the larval stage of the chironomids. I don't think my samples have anything like dragonfly larvae. I would have seen them. They're usually bigger. But who knows what else we might find. This is my first time actually looking at these samples. We collected material from this lake before. The last time I taught this class, we had some people that collected them. But I didn't do any kind of a live streaming or anything back then. And I had microscopes, but the students had all the samples they had collected, and I thought ahead this time and actually collected some samples for myself while everything was still alive, so we could get some images of it. And what better way to do that than to just go ahead and stream it onto Twitch, because then it gets recorded, so it's nice. And then my daughter's drawing stuff in the background behind us, and that's her working on a bird photo in Procreate that she's making. So she's 8 and working on, I guess she wants to do artwork. I wanted to use this time to point out that as well, that I think we're going to do another giveaway, some sort of a Redbubble giveaway, and we're going to do a coloring contest. So my daughter's drawn some things, and I think she's going to pick one out for us, and then there'll be uncolored outlines. And what we'll do is I'll post the coloring stuff in our Discord. Somebody's pushed a button. Yeah. So I'll post. Did that shake work? Oh, it did. Okay. My daughter is better at digital than I was at her age. Well, she's been practicing for a while now. Still working on trying to figure out how to space her name correctly in a sentence, though. Hey, Dangling, how's it going? So what we'll do is I'll post something in the Discord, which is here, and it'll just be an outline that my daughter has put together from something that she designed, and then we'll have a coloring contest. I will know who posted stuff because you'll send your submissions to me, and then I won't share that with my daughter or my wife, and then they will do some judging, and I will average their 2 scores to figure out who the winners are. And probably what we'll do is we'll give away, I have a a mustachio t-shirt that is slightly off-center. So we'll probably give that away, and then I'll give away some Redbubble gift certificates, and you can use them to buy merch for the store from the Diatoms Attack store if you want, but you can also use them for whatever else you wanted to. You don't have to use them for my stuff. But you could get stickers or t-shirts or whatever with it, or just use it to, buy whatever you want from there. For the winner and the runners-up, basically. So if I have enough people enter the coloring contest, then we'll have runner-up awards as well. So I think that will probably start after today, and then I'll run it for a month to give people time to do their drawings. You can color it digitally, or you could color it in pen and ink or marker or crayon, I don't care, color pencil, and then scan it back in. However you want to do your drawing, you can, and then we'll just judge based on how it colors. So this is a cool little cilia. We hung out on it for quite a while. What are you working on back there?
Yeah, remember the thing I drew you? Or no, not drew you. The eyeball thing.
You drawing an eyeball thing?
Remember the Cyclops pom-pom thing that I
Yes.
drew? I'm drawing him.
Oh, okay. Well, these little guys that we're looking at are also cyclopses in a way. They have one eye. That is a copepod nauplius, zooplankton, and this thing back here is a rotifer carrying an egg, and that's also a rotifer swimming through our field of view. Actually, maybe I should turn off the magnification ×2 as well while I'm here. I think I want this button. Turn that off. And then will be a little bit easier to see things all at once, because there's a lot going on in these samples. And then when I see them, you'll see them as well, because it'll give you the entire field of view inside the microscope. There's some Denobrian there in the center, and I still haven't figured out, and I don't know if I will, what this algae is that's around everything in the background. This little copepod nauplius. So copepods have a staged lifestyle, and they'll start off with the little nauplius. It'll molt, and then it'll grow and molt. And hey Evan, how's it going? I think it molts 6 times before it switches to the sort of torpedo with antenna body plan, and then it'll molt another 5 times before it's an adult and can replicate like full-size adult. Yeah, they do sort of teleport around. That's a normal thing. When you say an egg, do you really mean just one egg? Well, it's hard to say. They probably are carrying around a pack of eggs. It's this thing I keep seeing here, and it's probably a clutch of eggs. So you can see they have like a little oval sac or something that's underneath their body. I've never seen this rotifer before, so I actually don't know anything about its ecology. I mostly look at benthic rotifers when I dig around and look at stuff. I've never really looked at planktic rotifers, and I've never seen this species before. So I don't know the answer to that question. It's at least one egg. Sometimes rotifers have pretty small clutches, and they'll just produce a lot of offspring, like one at a time. So it's hard to say. This is a rotifer up here as well. This thing that has the little, legs that are sticking, tail spines that are sticking out like back end here. And you can see its front end still has the corona spinning around that it's using to pull itself around in the water column. So, or in this case in the slide. teleporting copepod Nauplius came through as well. Everything here looks like fourth dimensional beings. Yeah, lakes are pretty interesting places. My students saw some of the stuff swimming around in the water after we pulled up the plankton net, and they were like, I'm never going in the pond again. But wait till they see some of this stuff, right? Because they all look pretty crazy. Pretty alien looking organisms that keep swimming through. And some of those copepod Naupliuses just bolt through here so fast. When I was looking around in this slide, there's some of those rotifers that are basically inactive or dead right there, so you can kind of see their body plan a little bit more. They're kind of like tulip shaped or something. I did see, I'm pretty sure I found a Daphnia in here somewhere that was kicking around, so I'm going to just swim around in this slide a little bit. There's another rotifer, some more Nauplius, and see if I can find it. There's a rotifer with a little clutch that's not moving. Also, that's something we could take a look at since it's not moving. We can zoom in a little bit. There you can see the egg. It may be an egg sac. I don't know, but it's it's not attached. It's not an integral part of the body, and it's carrying it basically on its tail spine or around under its tail spine. So it's probably that's probably what it is. This thing down here with the radiant colony is a diatom colony. That's Asterianella formosa. Asterianella formosa puts itself in this sort of star shape, and then it uses that to help sort of baffle its descent. Its shape is basically like a Q-tip or something, but when you put all the Q-tips together by the head, basically you can make this sort of star-shaped pattern where that's where it gets its name from. So astero means or aster, it means star. So Asterianella formosa is a star-shaped diatom, but you wouldn't be able to tell what they were talking about if you only found one of them. If you didn't know what the colony looked like, you'd be like, why do they call that thing a star? It just looks like a Q-tip or a club. Do you think we can find these eldritch horrors in small ponds? Yeah, these are, well, they have to be deep enough to get plankton. So this lake actually, its deepest point was about 50 feet, but it wasn't very big. Elfda says, this is heckin' cool to watch in real time. I usually get my microbe fix from Journey to the Microcosmos on YouTube. This is neat. Thank you. I don't always stream living material. This is sort of in the evenings on the weekends. Sometimes I do. But I might be doing a little bit more of it a little more frequently because my class schedule is getting a little bit lighter now. I'm a full professor and I'm teaching 4 classes this semester, so I haven't had a lot of evening time. Just let yourself right in there, rotifer. Look at that thing just sort of butt right in, photobomb us. And hello, Alt Miss Frizzle. Thank you for coming back. Your students have bought you some cool samples from stagnant river waters. Yeah, you can find stuff. It's surprising, and I never know what I'm going to find when I'm looking in a lake. I do know if we took some temperature readings. We had a lot of sort of over the weekend. We had a whole bunch of limnological tools with us, so we could collect water from under the surface without mixing any of the surface water, and we could measure the temperature profile of the water column, and we were able to collect samples of plankton, and we collected stuff off the bottom. We collected cores, and my purpose for it is to have the students get some exposure to limnological techniques, but they each have to come up with a sort of a research project associated with their final grade for the class. There's a cool one, and he's just sitting there. You can see very clearly the front end of this one now, and you can see the cilia on it. So when they sit still, it's a lot easier to see the cilia, this ring of little hairs that run around its head. This is the anterior end, and it's spinning those little hairs the same way all the other ones were to eat and to draw food towards their mouth, and then you also spin those to help them move around in the water column. So I haven't seen any hydras moths. I've never found a hydra anywhere. I really wish I could figure out where one is, or where we have some sites where hydra occur. I know they mostly live in macrophytes or macrophyte areas, so there's a possibility something here that I have might pop out with one, but I have no idea. Let's see. Retention pond, anything. Yeah, any water places, you can find all kinds of cool stuff. Moth says, have you ever made ecospheres, like collecting water in a jar and watching what happens over time? That's actually something that Del Maximum does all the time. So he's another streamer here on Twitch, and a friend of ours, and he's done that several times, where he collects them and then keeps them in a jar, and then keeps the ecosystem going. These won't last very long. The plankton have very high oxygen demands, and I think it would be a challenge to keep them in a tiny jar. They would probably they're going to burn through the oxygen, and they're going to have a hard time in this setting. But benthic stuff probably you could keep. If I had a culture tank, I could probably keep the phytoplankton alive a little bit more, the zooplankton alive a little bit more readily. So, and Del's a mature audience streamer, and he does, sometimes he just, looks through crazy things that happen in the news, but he also often will pull out a microscope and start talking microscope stuff, so highly recommend him. I'm just going to move around a little bit, see if we can find something else. I put it in the corner because I'm trying to search effectively for some corpses of things. There's a Daphnia or a Bosmina in here. Oh, there's a Daphnia right there. Here we go. This is what I was trying to find. Okay, we're in super close though, so I'm going to move us out a little bit. I didn't put a cover slip on this because I was worried there was going to be Daphnia in there, but you can see this is clearly a Daphnia. And they're sometimes called, I think water fleas is a nickname for them, but people call all kinds of things water fleas. They're characterized by having a long tail spine, or a tail spine at least. The longer it gets, usually it's to avoid predation. And then they have a compound eye, and they have these swimming appendages that come off the front end of their body that make them look a little bit like zombies, like they're trying to eat your brains. But they're probably just looking for things to consume in the water. This one's struggling a little bit. But the compound eye is very distinct in Daphnia. It's a black center eye and then a whole bunch of little tiny eyes that are around the outside edge. The other thing that's really distinct for Daphnia, their body is divided, and the carapace actually has an indentation, so the mouth is right there, where I have the arrow, and the head has a little sloped head, and it's connected to the carapace, and then inside the carapace it's kicking its legs and stuff. And it has a big foot that it sticks out the back end right here. That's this shape you can see. It has a really big foot, and you can put it outside of the carapace here. So for identify them, the compound eye and the fact that their head is distinct and they have a little tiny snoot of a nose or mouth here, separates them from other things that you might find in the same system, like Bosmina. Bosmina are similar. They have a similar body plan. They're usually a little bit smaller, and then they have a trunk with a sort of proboscis-like thing that comes off of their head and splits in two. So it's a pretty cool image. This one is probably unhappy because the water on the slide is not very deep, and they like to use their appendages to stay afloat in the water, and it probably has no idea what's going on. So my guess is it's panicking a little bit. Their carapace, the inside legs in the carapace, it sort of has a slit in the bottom that can be wedged open or closed. So your lab used to keep Daphnia and C. dubia. He loved feeding them algae, yeah. So these are probably a little bit easier to keep if you have some circulating water to oxygenate it. And the legs are basically like inside the fold of their body, their main part of their body. And it's like I said, they can kind of put a foot out through the bottom. Think of them sort of like pants, basically, rather than like a hard shell that they couldn't open. So this is one of the common types of zooplankton. These are organisms that eat phytoplankton in the water column. So we did it before airplanes. Yeah, exactly. They've got landing gear, and you can see a lot of the detail on the skeleton. And if it would sit still like this, we can get in here and take a look at its head a little bit. You can see the compound eye structure. As I mentioned, it has like a black central eye and then a whole bunch of little eyelets that are around the outside edge of it. And those are used for detecting where the light is around them. Daphnia like to be in deep water, and they use the little eyelets to basically figure out if there's a dark area in the water. And their assumption is if there's a dark area, that is a shoreline. And they've run a bunch of experiments with Daphnia where they put them into lakes and in shallow water, and what they found is that they could detect, because of the angle of the light coming in, they could detect that there was a shoreline on one side, and then they would swim away from it and move themselves into deeper water. So this one's probably a little panicking. It probably thinks it's trapped in the shallow water because it is in shallow water, but it's trying to figure out how to move. I think they do have a heart. People run these really terrible experiments where they add caffeine to in biology classes where they add caffeine to the slide. Yeah, we were just looking at its eye, but then its heart's in there somewhere. Yeah, right there. You can see the sort of dark thing that's pulsing. Its eye is the dark spot in the center of its head, and in its chest, or the carapace, the main part of the carapace, you can see another dark spot in there that's pulsating. And yeah, so they run these really terrible experiments where they put caffeine, because caffeine is a poison. We're too big for it, and we take it in such small doses that it doesn't kill us, but it'll kill a Daphnia. So how many cells do these have roughly? Probably billions of cells. This is not a single-celled organism. This is a complex organism. It's a cladoceran, so way up the food chain from the single-celled stuff. We're looking at it at my second lowest magnification on this scope. This is a 100 times magnification. You can see these with your naked eye. So you can see them in the water. They look like little specks, but they're probably the larger sets of specks that you can see. So it's not it's a microscopic organism, but it's right at the border between what you could see. Like I said, you can see them as specks. You just can't tell anything about them very easily, without a microscope. So yeah. I mean, I don't know. I guess they feel like because they can replicate pretty quickly, you're allowed to do whatever you want to them. But I mean, I would prefer that they didn't do experiments like that. I mean, here we're looking at them, and I probably don't have any way of disposing of these in a positive way. But I think I'm going to try and record what we have, and then I'll try to dump them into my koi pond and see if my goldfish will probably eat them before too long. But that's a better outcome than just treating them. Yeah, it's an animal. It's definitely in the animal group. Pretty neat little organisms. So a couple of things about Daphnia that are pretty interesting. One, the tail spine, it grows longer when fish become active in the water and organisms that might eat them. And so they have a process called cyclomorphosis. They change the shape of their head to become a little bit more pointy, and their tail spines get even longer. And they use this to try to deter organisms from eating them. And then another thing that's kind of cool about the Daphnia is that most of them, and part of the reason people can have them in biology labs pretty easily, is that they every time they molt, the females will they have eggs that they'll form inside their body, and then they release eggs, and the eggs are just a clone of the actual organism. So most Daphnia that you encounter will be female, and they'll make the eggs inside their body, inside the carapace. They're stored usually somewhere like in here, and then they're parthenogenic, so they'll just make clones of themselves with a set of 4 or 5 eggs every time they molt, and they release those eggs out into the water column, and they'll just grow to be female Daphnia again. And then they have this weird broken wheel kind of a cycle of parthenogenesis where occasionally they'll conditions will get bad or weird or unfavorable, and then they'll produce male eggs, and the male eggs will then grow to male copepods within a few days, and they'll have regular sexual relationships with the copepods, and they produce something called ephipia, which are like seed bank eggs that they carry around, and then when they molt, they just go into the sediment and they wait until conditions get good, and then the seed bank eggs basically will spawn amictic female Daphnia again, and they'll return to producing offspring that are just clones. So it's a pretty interesting life cycle. Oh, I see. You're having some conversation aside from this. You're asking me more genetic questions. I don't know whether they're haploid or diploid, Dr. N. That's I think probably something you could Google pretty quickly though. So here's the little copepods. We have Daphnia that we've seen in here. We have the rotifers that we've seen, a bunch of different types. This is like a Caratella type here, the one that looks like a flower in the background. It's just a dead organism. What's going on, Sylvia?
It's almost 8:15.
What's going to happen at 8:15?
I go up to bed.
You go to bed.
Or something.
Are you done drawing? Because I have a white box up here instead of you drawing stuff now.
I've made this.
That's pretty nice.
And I've made this.
That's a mustache version of a puffball, right?
With one eye, so it's a cyclops.
With one eye, a cyclops, mustached puffball. I like them both. And nice job on the bird. It's got some pretty nice coloring.
I used reference image, but not for the colors.
Oh, okay.
And not for the patterns.
Did you make a decision on what we're going to have for the coloring contest this month?
Yeah.
Is it that frog drawing you did?
This one.
Oh, this is going to be the coloring contest image?
Mm-hmm.
Okay, so we're going to take that and we'll post it in the Discord, and then people can color it, and then that'll be part of the contest. It's relevant, okay. Oh, a screenshot you got from my stream earlier. Okay, it's fine. I mean, whatever. Xenophyte, have you ever seen freshwater sponge? Oh, are you done, Sylvia?
It's 8:14.
Okay, I want to turn off the view of the iPad then, if that's fine with you. Okay. So here's a little copepod. This one is, I think, a calanoid copepod, and it is still alive, but it's not very active. It's probably reaching some sort of state where it's unhappy. You can still see movement of it's either digesting stuff or something like that, the inside of its body. And then for these, you can see the little red eye here. Here's the long appendages that come off the front end of their body, and then they have a set of legs that are underneath their body that we can't see because we're looking through things on the light microscope. So, and I'm going to zoom us back out to full size. There we go. Have you ever seen freshwater sponge? I've never seen a living freshwater sponge, but I've seen a lot of freshwater sponge spicules in lakes. And in fact, every lake that I've ever looked at the sediment from, there are at least some freshwater sponge spicules in the sediment record, which tells me that freshwater sponges are, one, very small, and two, inconspicuous, probably. Hard for us to identify easily, but they're definitely there in all of them. And Moss Per Hour, your question about jellyfish, there is a lake here in Indiana, in Sullivan County, which is the one just below where my university is, and they have freshwater jellyfish in it. And also Lake Tanganyika, which is a lake that I do research in as a scientist, mostly the fossil record, but the modern lake has endemic Cnidaria as well, that are part of a project that we're trying to reconstruct. We put in a grant to NSF a month ago. I was really busy trying to get grant stuff together, and we think we can reconstruct the history of the jellyfish from ancient DNA in the sediments. So you would love to see a freshwater jelly on stream. I don't know. I think it's on some farmer's property, but there are definitely instances of it, and I'll see if I can figure out what lake it is, and if they'll let me come collect it, I will try. I think more commonly the jellyfish occur when lakes become stratified and nutrients become very low, and the lake becomes somewhat anoxic, because those anoxic conditions are usually jellyfish conditions in the ocean as well. And so I think that more likely they are a summer organism than anything else, but I'm not totally sure. So my expectation is that they're probably a summer organism, but I'll be around in the summer, and I'll see if I can get some. This little weird globe that we're looking at is actually Volvox. That's a type of green algae. Each one of those little things you see moving around in there is a flagellated, it's a colonial organism. And if I were zoomed in really closely, I think in some of them you can actually see they have a little red eye right here. There's one with a little red eye. You can see some of them in here that have a little red eye. And occasionally I think you can actually see the flagella moving around. But the Volvox are one of those things where they basically act as a colonial unit together. So they're a bunch of single-celled organisms that are living in a colony of, with their flagella, and they only occur in the colony. How are they holding? Yeah, they have like a weird globe shape that they live together, and there's some sort of connecting pieces between all of the organisms. So they're not free-floating in that shape. There's actually like a network of something that they exude from their bodies, so probably EPS, and it keeps them in the globe shape. And in fact, when they make daughters, the daughter colonies form inside the parent colony, inside the globe. And when they get big enough, what happens is the outer adult globe ruptures, and the daughters will come out of it into the water column and float around. So one of the things that's weird about Volvox is that they behave like almost like an actual organism, like a larger organism. They work together to swim in direction, to follow light sources, and Their reproduction behavior is almost like a larger organism's, in the sense that, like a multicellular organism, in that they produce their offspring, and then in these little colonies inside of themselves, and then they basically hatch by rupturing themselves. But that's a normal behavior for algae, and and so they have a really really bizarre, like almost multicellular component to them, which is really freaky. And here I'm trying to see if I can get the flagella at the field of view. So by putting it right on the outside edge out here, you can see their little tails waggling around. So each one of them has a flagella that they use to try to position the colony of the actual globe and keep it afloat. So super, super cool. Let's see. Moss Per Hour says, I used to be involved in a freshwater sponge project helping to ID the Field Museum's specimens. It was interesting. Yeah, so I've never seen a freshwater sponge in the lake, but like I said, I've definitely seen evidence that they're present, and their spicules are pretty common. So the spicule, if you're not familiar with sponge biology or ecology, they have a toothpick-like structure inside their bodies that are microscopic. They're called sclerae, and they will use those for defense. So they create a bunch of these little nasty silica toothpick things, and stick them inside their body so nothing will eat the sponges. So sponges are pretty pretty safe from other organisms. Their shapes are basically mush. Well, that's probably why I can't find them. So anyway, this is cool. I've actually seen Volvox a lot in videos, but I've never collected a sample where Volvox was in it before, and I didn't know where I should be looking. But you can see there's bits and pieces. This one I think is a ruptured one, but they're present in the sample kind of a lot, which is crazy to me. But I guess when you have something present, you always have something present in a lot of numbers typically. So you can see this one is working hard. What I love about this image, you can see that the organism is spinning the cilia like crazy fast, right? And you can see the particles moving towards its face. And you can also see these, like a separate little ring back here on the backside that it's waving around. That's sort of keeping its position a little bit, but they look very, like, I don't know, sort of like hair looks when it's underwater, just floating around the bodies. I think this is a ciliate, because I see a ring of cilia on the bottom part of its body and a ring of cilia on the top part of its body. I suspect this is something like Strombidium, which is a planktonic ciliate, but I don't know my ciliates very well, and that was a rotifer that just swam through. So you can see this thing actually has, it looks like it's attached itself to some piece of the algae here to keep from swimming away, which is why it's sitting in place. I don't think that's a stentor. It could be. It does have a stentor-like body, and ciliates. Stentor is a type of ciliate, but it's not doing the normal like trumpet thing that stentors usually do. So I think it's more likely Strombidium, which is a type of planktonic upper water column ciliate. I think it's this. I think that's how you spell it. No, Strombidium. Yeah, I think that's it. You've already exhausted your. Yeah. So stentors are actually fascinating to me. I really, really love finding them, and there's one in my koi pond in my backyard that we got on stream last, I don't know, maybe like 3 weeks ago or 4 weeks ago. I did a little evening stream. But it's in a similar family to those. But you can see that it doesn't have the little toes that rotifers usually have on the back end that are holding. It would normally be holding on with toes if it was a rotifer, and this thing only has like it can't tuck the corona inside its body the way that rotifers can. So you can tell this little ring down here of cilia, plus the ring that's up here around its head, means that it's not a rotifer. But pretty difficult to identify a lot of ciliates. I have a protozoa, Strombolium. That would actually be delicious, I think, if we could make a ciliate that was out of pizza. I only know Stentor because they're genomes. Yeah, I know. That's okay. You can be in your genomic world. You know, there's people out there that do that, so I don't have to, which I think is great. So this is a cool little guy just hanging out here, attached to some of this algae. So this is most of what I've found in the living samples. Here's another. That's a little cluster of that is not that brown clump in there is not a Volvox. It's a clump of that's green algae, I think. It's just or cyanobacteria maybe. It's a little coccoid colony. I think that maybe we've seen all the organisms that are moving around in this sample. I did want to, I had made a slide where I just put a cover slip on it so we could kind of get close to some things. And I wanted to look at that before I move on to the benthic stuff. And so I'll see if I can figure out what I did with that. Hopefully it's not completely dried up. But there was a collection of material that I took off the bottom of these centrifuge tubes that I'm using. You can see, like, hopefully you can see down here at the bottoms where all the dead stuff is. And then the organisms are actually, they're still swimming around up in here. I don't think you can see it very well from the tiny webcam, but trust me, that anything that's stuck in there that's still floating around, it's not clear water, is because it's maintaining its position by floating, or using some sort of a cilia or a flagella or swimming appendages to stay afloat. And then this sample is taken from the material from the bottom so that we could look at some of the other organisms that are present. These were probably living when we started, but were collected early this morning, so the organisms are probably not alive anymore. And they were collected from the bottom of that little vial. But this is Bosmina, and as I mentioned, it has a similar body plan to the Daphnia, but here you can see it has like an elephant trunk spine coming off of the front end. It has the same zombie arm situation up here. The thread things might be cyanobacteria. I don't know what those little thread things were, and I didn't look closely at them. I got the sense that they might have been Phanizomena or something like that. They're here as well in these little clumps. I'm hoping my students will actually somebody will take up an interest and try to figure out what this little clump of stuff is. So it's probably cyanobacteria, but I don't know for sure. That's the corpse of a copepod right there. Here's a dinoflagellate. This is a Nauplius corpse. You can see there are some large copepods. What's nice about this is that we're actually getting to see the copepods. This is a calanoid copepod. It has a much larger body plan, but you can see the very long antenna that would have normally been in its living position sticking out the top of its head like little horns. But you can also see all the little legs that it has. And the reason that they look like they teleport when they move around is that their legs have little like paddle-like structures on them. And then it has like a little caudal piece back here on the back end, which is where, again, it keeps its sex organs and stuff like that. And if it had eggs, it would keep them associated with the edges of the tail end of the organism, the posterior end. But this is nice because you can actually see all the swimming appendages down here at the bottom that they use to sort of kick around in the water and its whole body plan is sort of visible, so it's kind of neat. Um, it's like a fossil impression, yeah. Well, it's on a mounted slide with a cover slip, so that I can zoom in if we wanted to and look at some of the stuff a little more closely. This is probably one of those ciliates right there. It looks like the bacteria is having its way with it. That's a copepod. Nauplius, there's a bunch of Nauplius in here. And the there's a bunch of Daphnia. No, those are Bosmina. I thought I saw another one in here. I thought I saw Chidorus. That's a Daphnia. So the way that you can tell, I'm sorry, that's a Bosmina. The way that you can tell it's a Bosmina, it has the long trunk, and it has the compound eye as well. So that's a very good impression of those. Good evening. What's going on? Strikacek. Welcome in. There's a giant Daphnia next to a tiny little Bosmina, and you can get a good sense of the differences in the size of them when they're like this side by side. So Daphnia are usually the smaller organisms. They have the trunk, they have the compound eye, they have the same, more or less the same shape, body plan. You get itchy. Sorry, Frankie. And then this is the Daphnia. Here's its little mouth, and the swimming arms. We can actually zoom in on, I think we can zoom in pretty close on this organism. Let's see if I can manage it. Yeah. Here you can see all the detail if I zoom in. So because I have a cover slip, I can get really close, but the mouth is right here. The compound eyes here. The arms, the swimming appendages are up here. And then I mentioned that they have a little foot that they can stick outside of their body. So they have one little kicky foot that basically can come out through the carapace, and that's the kicky foot, has little spines on it, and I think that their intestines are linked through that little kicky foot, so it's also a way for them to get their stuff outside their body. So pretty cool to be able to take a look at this stuff. Oops, I don't want to do that. We're going to zoom in and look at the actual little foot spine so you can get a nice clean look of it. And then this is, the foot of the organism, or one of the feet. And like I said, it's linked. This is the intestine sort of here coming out the foot as well. So yeah, it's like a giant claw. Exactly, Moss. Alright, let's zoom back out to the 10x. When I'm in too close, basically I can't see any of the details. I really just wanted to see if we could find some other stuff in here. You can see, if you look right there, you can see something swimming around the outside of these things. And normally the progression is when zooplankton die, bacteria start to take over, and also a lot of the little ciliates will show up because they are capable of decomposing things. I mentioned they can swim around with egg sacs being carried around on their tail ends, so this copepod, single eye, a bunch of little legs under their body, and then you can see a whole stack of little eggs this thing was carrying around. Unfortunately, those eggs are not going to make it. I'm afraid they were not successful, but they're clones, so don't worry about them too much. Think of them as the stormtroopers in the Star Wars movies. Not that they're bad guys, but that they're expendable. So there's another copepod. These little Nauplii everywhere. There's a diatom that also got trapped in our slide collection. Here's one of those Caratella-like rotifers that are here. Yeah, she had some pretty big looking eggs, didn't she? That's probably why they carry them where they do. So that's a rotifer. The spinny end is up here. This is the anterior end, and then these are the tail spines on this thing. I think it's Caratella or something like that. They're predatory rotifers that live in the water column. Um, oh, could we go back and zoom further into the eggs? Sure. If I can figure out where I left them. Oh, here's some eggs that are just out hanging on their own. Uh, you want to zoom in and look at these ones, or did you want the ones that are attached to a body? I think these are the same type of things. We can get much, much closer. This is the, like I said, we're on the lowest setting on my microscope, so we're looking at some fairly large stuff relative to diatoms, which is what I normally look at. But we could get a lot closer if you'd like. We can zoom in and look at them on the inside. I don't know that they actually have a lot of structure to them. 1, 2, 3, 4, 5 little circles. I don't know what those are. These work, yeah. Okay. Yeah, I don't know. I can focus through them. You can kind of get a sense that they're spherical. But outside of that, I don't know what else we can tell. There's also a lot of interference because I have the differential interference contrast up super high. So I'm going to change that. I'm going to change it to more like a bright field appearance by turning up the brightness and see if that helps. Well, it does give us a little bit different appearance or a little bit different view. You can see what's going on in there, right? A bunch of those little spherical cells are more obvious when I turned up the brightness. So you can see a little bit more of them when I don't have the interference contrast so high. Sure, no problem. Whatever people want to look at, it's cool with me. I'm just kind of exploring. So, again, that's what they look like without the heavy contrast. And I've been running it on the other end of the spectrum in the heavy contrast because I think it just looks better. Have a darker background when you're on camera. There's a bunch of those little rotifers. Looks like they were having a powwow. Now permanently having a powwow. Daphnia. We saw this cluster. I swear I saw a podon in here. That's a copepod. That's a bosmina. I swear I saw a podon somewhere as I was moving around in here earlier. The skeleton of one. It wasn't moving, but that's a copepod. Nauplius, copepod. I think we saw that one. Yeah. That's why I usually try to find some sort of path and I stick on it so I don't end up going back over the same spots. Copepods. You can see they're actually quite abundant. Copepods have sexual reproduction lifestyle. They don't have the parthenogenesis. But rotifers, many of the planktic rotifers actually are parthenogenic as well. And Daphnia, bosmina, Chidoris, all of the Cladocera are commonly have a parthenogenic lifestyle. So here you can see the differences between things again. There's a little beak on the Daphnia. Here's its foot. Here are the swimming arms, swimming appendages. Here's the big long tail spine. Oh, there's a, that's interesting. It's a little dinoflagellate that I didn't see living in any of the other samples. As I mentioned, I've seen a podon in here somewhere, and I also found Chidoris. Those are podon is a type of Cladocera. It just has a different appearance. It has one big eye in the center, and then Chidoris is also a type of Cladoceran, a little less common. They have a little bit more of like a tiny triangular head. but I'm not seeing any of them right now, so it might have been in another slide that I had made, because I just sort of went through a few of them before I started. There's another one of those different types of rotifers right there that's kind of drifting around. Okay, well, let's go explore some of the benthic stuff and see what we can find. So these are just samples from plankton that were collected in the lake while we were out in the field today. I asked the students to grab me some, and then I proceeded to grab some of my own samples a little bit later after everybody had left, or I'd taken everybody home. I collected some sort of scuzzy stuff off the top of a swamp. This one is some lemna, some duckweed off of the top of a swamp area. It's more of a wetland than a swamp. And then this little bit came from a boat ramp. And then I have another little piece that came from a boat ramp. And they were in the algae that was growing on the edge of it. I was hoping to see what kind of diatoms we might find, but who knows. So far we've only seen Asterianella formosa in the plankton, and then this thing I collected from just a part of the lake where there was a bunch of little algae in a sort of grassy area where there was a bunch of frogs. So I just wrote frog on the sample so I can remember where it was. And I think we can probably put a cover slip on this because there shouldn't be much plankton in these samples. They should just be stuff that was living in the macrophytes. So it could be anything in there. Again, I haven't looked at these samples at all. So there could be anything. It's a mystery what we'll find. Could be something exciting. Could be totally boring. This looks like Oligosyra. That's a type of diatom. There's a bunch of ciliates in there. There's also a bunch of little diatoms growing on it. That's a piece of plastic. I'm going to take this as my public service announcement. That is a piece of plastic that we found. It's blue and fibrous. There's always plastic in everything that I look through, by the way. That's a message from my friend Asymmetrical Soul popped up. I think I'm with whoever said that they're never going to lake again. These are little organisms. They've always been there. They aren't going to do you any harm. They're little. And just don't drink them. They probably don't taste great. Yeah, all the mouthful of weight. Yeah, you know, the thing is that a lot of times the plankton that we look at, it's been highly concentrated. So you might get some stuff in there in your samples, in your mouth, but it's going to be relatively limited. It's not going to be like a mouthful of plankton is probably not what you're going to get. It'll mostly be water. This sample is all diatoms. There's a whole stack of diatoms right here. These are a whole bunch of them living in a zipper colony, a ladder colony. Probably Fragillaria, and then all these little things are all diatoms. Like everything you can see here is diatoms, basically. Except for this stuff. I think this is Cladophora. This like long stringy stuff that is not regularly spaced material. So this is nice in the sense that it let me definitely will have some diatoms in these materials. Oh, here you go. There's a little worm. Is it a chironomid? We'll come back to it. It's nestled into the middle of all of the junk, so I can't get a bead on its head. And I wasn't able to pick out any of the normal chironomid features that we see on their legs either. This looks like Oligosyra, maybe. And there's a huge zipper colony. Look at that, like a ribbon of diatoms. Very pretty. So this sample will be nice to look at in the scanning electron microscope and in the prepared samples after we get rid of all the organic-y junk. Oh, what was that? Hello? I think that was an ostracod. Ostracods are kind of related to the Cladocera arthropods. So let me zoom out so you're not like constantly wondering what I'm talking about. There it goes. It zoomed down here somewhere. I don't know. It might have gone outside of the slide, or it might just be taking laps. That's a rotifer. Slow-moving rotifer. What do you want, Wednesday? That is our little worm. It's not a chironomid. It's an annelid. Oh, it's a Chidorus. Yeah, it's not an ostracod. This is the thing I saw. There, you actually saw for a couple of seconds there they have like the inside of their shell. So I was telling you, you could kind of see that they were a little bit like pants, that they could stick their legs out. Earlier in the stream, we were looking at something that wasn't. This is Chidorus. It belongs with the rest of the Cladocera. See, it's got a compound eye, but instead of having the typical Daphnia arms sticking out, these are a little bit more subtle. But it still has the very distinct head there, and sort of like a Bosmina trunk almost that you can see. So this is Chidorus. And you can see it's kicking its legs the same way as the ones that we saw before, right? Pretty cool. It's a spaz. So this is Chidorus. I don't know where it went. It just zoomed out of my screen. Wednesday. Okay. My cat wants to get a chance to look at what's in the microscope. Up here? Is this what you wanted? Prime time? You want to look in there? It's not as interesting as you thought. Okay, get down. If I don't pick her up, she just sits down there and paws at me. Let's see. Stricasec, nighty night. Good night. Thank you for following 990. And funny, the first species that discovered how to use microscopes became the dominant species on the planet. Well, we became the dominant species way before we learned how to use microscopes. But our domination is not in numbers. There are many fewer humans than there are Cladocera on planet Earth, for example. Nor are we very speciose as a genus. So it depends on what you consider to be dominant. There's a lot of other organisms that are way more common than humans. We have a lot for vertebrates. I think we're the most dominant vertebrate on the planet, but we're nowhere near the dominant organism if you're going by numbers. If you're going by warfare power, we're pretty good at that stuff. But I don't know that the microscopes are directly linked to our superiority in any way. There's this little worm. That is an oligocete, I think, or a nidid worm. Nestled in here. Just a little too zoomed in. Let's see if we can get him in our field of view. There he is. Got a little a hairy mustache on that one. Not even one that I put on him, just an actual hairy little mustache. And you can see into their body. Most organisms in freshwater systems are translucent or transparent. The pouch their legs are in reminds you of the little rubber coin. It almost exactly like that. You have to sort of squeeze the shell in order to get the legs out. They are like a little coin purse. Exactly, Evan. and then the legs come out instead of coins. So there's a direct link between Twinkie consumption and all superiority. That could be, that is a good point, Disillusion. We do eat Twinkies. But you can see little hairs right here. If you look very quickly, there's little pairs of hairs coming off of segmented bodies. So that's an annelid, a worm, and the hairs are usually used to help them grip what they're moving through. Sets of pairs of things like that, pretty common in the bodies of worms. This little guy, I think, is a Vorticella. I'm going with Vorticella for now because it looks like it's attached singly. And if I get the focus just right, you'll see it has little cilia surrounding the top of its head as well. This is a type of ciliate. And this one we find regularly. If you're a follower of my friend Studio Cornix, she has some very beautiful artwork associated with these little guys. Here they have a bell-shaped, like a flower head. This is a type of ciliate, so an attached version, and then up at the top a crown of cilia that run around the outside that allow it to spin and pull food towards its mouth hole. And then the Vorticella live on a single springy sort of attached component that they can pull themselves back in, like a little turtle. They can pull themselves back in towards whatever to avoid things eating them. And these ones are living side by side. So the halo that you're seeing is actually a little tiny set of hairs. And I'm going to zoom in. I'm first going to zoom out, and then I'm going to zoom in on it with the 20x magnification. And you will see that those are little hairs instead of, hopefully, you'll be able to see them. On these guys. So there's two of them in here. They're living basically side by side. It's not a colony. They're attached independently. But let's see if we zoom in really closely at 20x, if we can actually see this, the cilia, the little hairs that are there. They're spinning super fast because they're using them to bring food towards their mouths. But the little halo that you see right here, those are hairs. Sometimes they'll slow them down and you'll just see them waving, and they look like actual hair underwater. They run around the outer edge of them. They look like they're spinning when we get kind of close to them, but they're doing this sort of a thing. They're sitting in place and they're just flipping and folding, and then as a result, when you look at them, it looks like they're spinning. So that's a good observation, Semi. Yeah, coronas are what we usually call it when they have a set of little hairs that go around the top of their crown, basically, a crown of hairs. So, but this is a ciliate, not a rotifer. It's attached. And you can see into its body again, very bell-shaped organisms. Let's take a look around and see what else we can find in here. This is a 20x, so we could even get closer. I'm afraid it's not moving, or it's moving a little too much for us to actually see a whole lot in here. If we were to zoom in any closer. If stuff is sitting still, the depth of field is usually the issue on the microscope. And so if stuff's sitting still, you can usually get a good sense of the depth of, or the keeping them in focus. But if they move and your depth of field is very small, they're just out of focus constantly. This is a little ribbon colony of diatoms here. You can see the diatoms are single-celled organisms here living in a colony. They're not moving on their own. They're being bumped by me, I think. When you say hair, do you mean setae? No, cilia. what's the difference between cilia is a little flexible hair that are found in clumps that organisms can use to move around or move food towards their mouths. And so they have like a purpose for spinning or for mobility or whatever. If there's just one hair rather than a clutch of them or a clump of them, we call it a flagella. But functionally, they're basically the same thing. It's just that a flagella, there's one, like a whip, and they're used a little bit differently. They're usually longer, and cilia are usually short and in groups. And setae are something that you would find on a worm that's like an actual hair that come in pairs that come off of the body. And they aren't mobile, they aren't flexible. They're like rigid hairs, like my beard hair basically. They don't use them to crawl or to move. So most of those designations are about how the thing is used or the sort of shape of them, rather than. Yes, this is a benthic sample cyanophyte. So the terminology is a little bit about how the organism uses it, and then the positioning and how flexible they are, and whether they're whether they're in clumps or individually. So yeah, they're kind of overlapping terms, but I never confuse them because I always think, well, that thing has just one of those, so it's a flagella. So it's like flagella means like a whip, and so you only call something a flagella when there's one or two of them and they're long and they use them like a whip. Whereas cilia are always in like bands or rings, and so I don't ever think of them as they overlap in terms of like function, and it's like a hair, but I think that they have different direct uses, and so I don't ever really confuse them, or if I do, I usually correct myself immediately afterwards because I think that they're pretty distinctly different things. You know, but the problem is, of course, that people just have they're microscopic organisms, and a lot of the names were put on things before people could see them perfectly, and then we just adopted those names. There's the Chidoris. So separate from the other organisms that we were looking at in the Cladocera group, Chidoris have two sets of eyes, and then their beak is this little tiny. This is their head, just this little beak and the flat part up here. So this is all their head. You see they have the same foot out here, and they open like a coin purse, as Evan mentioned. And usually they have a flat bottom instead of the tail spine that's sticking out like a Daphnia. And internally they're pretty similar. They have the same. There's a heart. There's their legs. There's this big foot that they use, and usually their intestines tied somehow to the foot. I think this one's carrying an egg. I'm pretty sure that's what that is, or a clutch of eggs or something, because I think they usually carry their eggs around on the backside up here. So pretty cool. I don't know why that info is just stuck on the screen. Stupid stuff.
Good news, everyone.
What about those things we saw earlier? They look like they had four or so squid kind of things. Would those be flagella? I think those are actually. I think what you're talking about are the rotifers that have these like long spines sticking off of them. Those aren't flexible at all. They're actually part of the carapace, and so I think those are just spines for those, because they don't use them like they don't try to whip with them. The only things that we've seen that have a flagella that I clearly recall are the dinoflagellates. The flagella's in the name, and then the Volvox had those little tiny. They just have like a one or two little whips that are associated with the cell. I did want to point something out. There's a really cool diatom colony right here. I love this. That is a colony of amphora or or Ensynema, one or the other. I think it's probably Ensynema. But they live in these little tube colonies. So this is a single-celled diatom and another single-celled diatom and another single-celled diatom. They're just living in a colony together. But they're like little cars in a tunnel, basically. They can crawl around up and down in the tunnel. And this is their chloroplast right here. So pretty cool. Piper Lee, hello. Hey, if you're not following Piper Lee, I think I made I gotta check. Piper? Is it Piper? No. Is it Piper Lee? Huh? Aha. I made a little special command for Piper Lee. She plays Irish music with the tin whistle and bagpipes, and she does loops and plays guitar, and very entertaining. We're looking at some samples that I collected on a field trip that I was on this weekend that I was leading. I think most of the time the music streams are on Wednesday, is that right, Piper Lee? So earlier my daughter was also here drawing some stuff on her iPad as well, and I wanted to mention again, if people weren't here for the early part of the stream, we're doing a coloring contest. Everybody knows how to color. Some people are going to do it better than others. Some people are going to pick colors that appeal to my daughter better than other people. And if you join our Discord, I'm going to post a picture of something that my daughter drew, and then you color it, and she judges it. And then my wife will also judge it, so that there's two judges in case my daughter goes nuts. There's no cost to enter other than just joining the Discord, and if you win, then I will send you either a Mustachio t-shirt with that slightly off-center that I got for, I bought one for myself, and it was slightly off-center, and I was like, why is this off-center? And they said, we'll just send you another one. So I have a free t-shirt that I'm going to give out to winner Sarah Dance Painter. Hey Sarah. Look at my automatic raid announcement, worked perfectly. I built a little code that does that when people raid. It sends out the announcement, and then it does a little who to check out what the person is. I was just telling people we're doing a coloring contest, so you can join my Discord and win a t-shirt. Or if you don't want the t-shirt, which people have different tastes, maybe you don't want a Mustachio emote t-shirt, you can also opt to get, I'll just give people some money for Redbubble, so you can use it however you want. You don't have to spend it in my shop, but you can spend it in my shop if you want. So, but we're doing that for the next month. A sneak raid. Sarah, what were you doing on your stream? Were you dancing and painting? You can't hear. Oh, okay. I'll give you a second. I think she normally dances around and does abstract painting, which is really cool. And Sarah used to hang out in our channel all the time. And then she stopped streaming for a while, and now she's back. So I'm excited about that. I think this is a little rotifer in here that I'm seeing. We're just looking around in some samples that I collected. There's another piece of plastic. I think it's a different one, but it's still blue. In this material that we were looking around in, which I feel like we've kind of exhausted what's here. But I will probably put this on the SEM in the coming weeks. We'll look at some of these materials in the scanning electron microscope as well. I'm going to make a slide out of one of the other samples. I actually collected a lot of stuff, and I kind of want to look through it tonight and see what's in all of it. I've only looked at a plankton sample, and there's this boat ramp sample, and this other boat ramp sample. I think this one's probably going to be a good one, so we'll take a look at it. See if I can get some of this to come up without taking all of it. And we'll see if we can find something more interesting. We're just kind of poking around in the samples. And seeing what we find. So hopefully you can hear again, Sarah, and then you can tell us what you've been up to. If you could take a sample from anywhere in the world, where would you take a sample? That's a good question. Generally, I can take a sample from anywhere in the world. I don't have a whole lot of things that limit what I can do. I really desperately would like to look at a sample from Grand Prismatic Springs, and it's one of the few places I can't take a sample from, because you're not allowed to sample there without a permit, and I don't think they just let anybody get a permit. I have worked in Yellowstone National Park before, and I've done research there, and I feel like I have some people who could probably hook me up if I really wanted it. But I was just curious to see if I could find any diatoms in it, because it's one of my favorite parks to go to. I've collected stuff from Glacier National Park, and I've worked with people also from Acadia National Park. But to get back to a sort of like what kind of sample would I get, I answered this question about half a year ago to Pacific Plankton or one of our viewers, and I mentioned that there's two things that I really want to sample, and they're not lake samples. One is a sloth, and the other is a hippopotamus. And I don't want to be there for the hippopotamus sampling because those things are dangerous, but I want a sample of one of them because they go in the water, come out of the water, and very likely they have colonies of algae living on their skin, and very likely some of them are diatoms. And then sloths notoriously have algae growing in their fur, so I kind of want to see what diatoms are living in sloth fur. So those are kind of like the out there answers that I have. But when I mentioned this to Pacific Plankton, the hippo might be a little dangerous. They tranquilize them, and they take samples occasionally for blood and things and tagging them. And she mentioned to a friend of hers, Steve Mandel, who always shows up in her streams, and Steve and I have a lot of conversations about stuff. And she mentioned it to Steve, and he's like, "Oh, I think I can get you a hippo sample." And he's working on getting me a hippo sample. So that's on my Christmas gift from Pacific Plankton, was like hooking me up with Steve, getting me hippo samples. And he also has people who work with sloths, and so I'm going to also get sloth samples. And it's on my bucket list of things to look through, and I'm going to get both of them probably in the next year. So I've got to come up with some new crazy ideas. But I'm excited about both of those, and we'll probably stream the hippo samples and the sloth samples if I can find diatoms in them. So I love hippopotamus. It's like one of my favorite animals. And I mean, if you had to pick an animal to like, hippopotamus is one of them. And I like them more than humans, that's for sure. And then, even though they don't like humans, I don't either. So we have an understanding. But what is in here? Something is wiggling around. Hello? Oh, that is another Chidoris that just, I think, that just butted itself into my sample. But there's something here crawling around in it that I wanted to see. I think that's a nematode worm. I think it's just a worm. I'm not positive though. It could be a Lacrimaria. Its head is wiggling around a lot, so it's either a Lacrimaria, in which case I'm going to be very happy, or a nematode worm, in which case I'll just be like, whatever. Nematode worms are everywhere. But we'll come back and take a look at that. You would love to chill and look at algae from a sloth. Yeah, exactly. What about that pink acid lake in Tanzania where only the flamingos chill there? So that is Lake Bogoria that you're talking about, I think. And it's mostly filled with cyanobacteria, Spirogyra, and and cyanobacteria. And hello? Look at those little arms. Look at its little arms. Look at it. Hello? Did you want to be in the light? Come on, dude. I'm trying to look at stuff here. Look at the arms. Look at it's wiggling its arms around like crazy. I don't know what this one's called. It looks like Kydoris, but I don't think that's what it is. Look at those crazy arms. Look at it's got a foot that sticks out the back like the rest of them. It's a Cladocera of some type, but I actually don't know which one this is. Oh no, it's a Kydoris. 2 sets of eyes. It just wasn't sitting still long enough for me to see. It's doing donuts. Hi. Is there something you wanted to tell the viewers? You just wanted to show them how your foot works and how your arms work, and you're very excited to be here. Okay, understandable. Yeah, he's got a lot of speed. Well, it's probably a she, but she's got a lot of speed. Look at that. Look at that. Crazy spin. This thing's like, it's very excited. Thank you for the follow. Yeah, exactly, Piperly. Just like somebody has a hot dog. This is spinning around. Look at it. Perfect. Yeah, it's a Kydoris. What a cool. I don't think it'll sit still long enough for me to get a good image of it. It is so excited though. Boop. Nope. You're not gonna. There we go. That's sort of what it looks like a little bit more. It's a little more clear when I zoom in what it's like for me in the microscope, so it looks a little bit more like I can see a lot more detail than it can show for you guys. Just because of the way the light works when I'm zoomed out. But there you can see its skeleton, its carapace, and that thing is just spinning like a top. Pretty cool. Yeah, it's some sort of Kydoris. What's this? Hello? There's a lot of stuff in these benthic samples. I think it's a worm? Hard to say. I think that's a worm. That on some level the plankton are often drawn towards the light, so we'll see them basically come towards the middle of the screen. There's a diatom for us. Diatoms look a lot better when they're mounted samples. There's actually a lot of diatoms in our field of view right there. This is a traditional benthic diatom shaped like a diamond or a canoe. Oh, there comes the Kydoris again. Hello? Yes, I'm trying to talk about other stuff though. There's some Fragillaria colony. This is, it's probably a star anise, but I can't tell for sure. And then all these little guys are diatoms. You can see they crawl around. Most people probably don't think of algae as swimming or crawling. Hopefully I've broken a little bit of that concept for you. They do like to, the diatoms, the benthic ones can walk around or crawl around like sort of snails and interact with their environment pretty closely. Looking for light or a place to attach. There's a little copepod in here as well. Oh, it's still alive. That's sort of surprising, given that I collected this out of off of one of the boat ramps. I think that's a ciliate. It seems to be spinning in a circle, and I'm not spinning it in a circle. I think we're looking down on it from the top, so the ciliated hairs are going around like this, around the top of it, but it's hard to tell what the shape is because it's a very small one. Just sort of spinning spinning itself around. These are cool samples. Like I said, I didn't get a chance to really look through the living material the last time we went out to this. We had this class, and I led a field trip to this place, and because it was about 4 years ago. The 2 years ago when we would have normally gone was COVID, and I had done all this planning to get people to come to the site and to figure out how to get all this stuff done, and then all field trips in school was canceled basically, so that sort of sucked. It sucked for the students as well, and it was really problematic because like 20% of their grade is this final project, and I had to come up with something that they could do instead. So like frustrating on every level. Field trips are fun, and almost all of the classes that I teach at the university have some kind of field trip associated with them. So it's the carapace of a Posomina. You see how we can take just a little bit of what we know and apply it to even get from fragments what we're looking at. There's the trunk-like structure, and then this thing's broken open, but you can see that the head piece is completely associated with the carapace as well. And if it had been living, the eyes would be right there. But we can tell what it is just from fragments. Cliff Alistair McLean, hi Diatoms Tech. Have some questions about the shapes of diatoms. Throw them at me. Any questions people have, feel free to ask. Don't feel like you can't ask questions about anything that's going on in the channel. Definitely, if you have questions about diatoms, throw them at me. I think I'm your best source for information on Twitch for diatoms. Unless some of my taxonomic colleagues have started streaming and didn't tell me. I suppose it's possible. It's like rotifers. There's a rotifer. Zooming around. What do they look like? Banana shape. The side view, circular. Or are they really banana shaped? SA fox molders, do they fart? No. They're single-celled organisms. They don't have a digestive tract, and they are autotrophs, so they don't eat anything. So the answer is no. For McAllister, Cliff Alistair McLean, I mean, when they look like a banana shape, is that the side view of a circular one, or are they really banana shaped? The problem with that question, Cliff, is that diatoms are three-dimensional objects, and sometimes you might be looking at something that in the side view looks like it's a square, and in the vowel view or the face view it looks like it's a circle. So if I asked you what's the shape of this thing, this roll of duct tape, you would say, oh, it's a circle. But if you saw it like this, what shape is it? Well, it's a rectangle, right? So generally speaking, you need to have more than one view to have an understanding of what they look like in 3 dimensions. In the light microscope, we're looking through diatoms. So you could see them in any possible direction, and they may look like a square, rectangle, or circle, or even a banana shape, and then in side view they'll look like a rectangle. So it may be challenging to try to tell without a scanning electron microscope. The scanning electron microscopes have they don't use light, they use electrons, but they give you a topography, and so you can actually see them as 3-dimensional objects. So got some diatom questions too. What are the most important industrial and medical uses for diatoms? Industrial uses, they're used as filters. And I would think most people would argue that the most important thing that they can filter is they're used for beer filters and wine filters. They're also used as pool filters. So as diatomite, as the skeletons of ancient diatoms are used for filtering stuff because they have these lacy skeletons that trap a lot of things, and they're particularly useful for things like wine filters and beer filters and pool filters. So things that you might like to do in the summertime, and so thus they have some importance in that role. In the old days, they were used. You remember, like the old timey people had sticks of dynamite that you would light and run away from or throw or something, and or have a really long fuse, hopefully. Those are diatomite packed into a cardboard tube with glycerin mixed in with them, and then blasting caps put on the ends of them. And in that case, the diatomite is used as a stabilizer to stabilize the glycerol, to keep it from interacting with itself until you want it to, which is what the blasting caps are for. It's used in toothpaste. It's used in just diatomaceous earth people sprinkle around their gardens, for example. So, next question from Sinophyte. What is the oldest diatom in the fossil record? The oldest not disputed age for diatoms, like everybody agrees it's a diatom, is basically the age of the dinosaurs. So they've been around since the Cretaceous-Jurassic intervals. And in freshwater systems, the oldest known one is from the Wombat Formation. It's some work by Pete Seaver, and I believe it's 82 million years. And marine record of diatoms is much older. It goes back to the age of the dinosaurs. So the records from freshwater systems are much shorter, much more recent, but still quite old. There's a recent, if you're interested in the really old fossil diatom record, there's a recent talk by Pete Seaver for the Diatom Web Academy. If you just Google Diatom Web Academy, or if you go to the Diatoms of the US webpage, you can click on the Diatom Web Academy and then look at the presentation from 2 weeks ago, and they store all of it on YouTube. So if you were really interested in the ancient stuff, Pete Seaver is an incredibly interesting guy, and he talked about a lot of his research and its relationship to the introduction of freshwater diatoms. So a pretty useful talk. Piperly, what software do you use to capture camera input? My husband wants to know. He's got a microscope and a camera, and he's been trying to get it going. The tool that I use is my camera. Let me show you. So this is a microscope here. This is a research grade microscope. It's the Leica DM2500. What makes it expensive, so it's about a $20,000 microscope. What makes it expensive is it has differential interference contrast, which is what we're using right now to look at our sample. But you don't need that for a fancy microscope. It has a phototube, which connects to an adapter. The adapter basically magnifies the image to try to make it appropriately scaled for the camera that's on here. This is connected with a C-mount because my camera is a mirrorless camera, and so I can use a C-mount to directly send the image onto the sensor for the camera, and then I just have the window, the screen of what you're seeing is up here. It flips over when I turn it, but and then this thing that you see coming out of the camera is micro HDMI cable, so it gives me pure HDMI out, and that goes into a Camlink camera card that I use to capture the image. So Camlink is I think maybe $60 to $80 or something for it, and it I can't show it to you, but basically it has a USB plug-in. So I have it plugged into USB 3 in the back, and this card goes from, or this cable goes from micro HDMI into normal HDMI into the the cam link. So camera has a mini HDMI out or micro, whatever, whichever one it is, and then it goes into the cam card, which is USB, that just plugs into the computer as a USB. And OBS recognizes the cam link as a camera, and so it's just coming straight out through my camera into OBS through that link. So hopefully that's helpful. If you're if you want if your husband wants advice about it, and he's having some trouble, feel free to join the Discord or have your husband join my Discord and post questions and show us your setup. And my friend Pacific Plankton also, and I both, she hangs out in there a lot, and if you ask questions about microscope stuff, the two of us can probably give you some pretty good explanations about how to set up your system. So we both have a lot of experience. I've got a couple of camera options that we've used and should be able to help. So feel free. Yeah, I'm being shaken up. Let's see. Cliff Alistair McLean says, so you had to learn how the same species look from different sides. Yes, I do. I had to learn, most diatoms land in a preferred direction. So to get back to your question, Cliff, if you set something down that's shaped like this, most of the time it'll land on the flattest side, but it could potentially land like this, so that you're looking at the narrow side, or it could potentially land on one of the other sides. This is a rectangular shape, but each of the sides is totally different. And but diatoms have a preferred direction that they usually land in. Like a coin has a preferred direction, but it might land on its edge. So most of the time, the diatoms that we see that are coin-shaped land so that the circular face is down, or if they're beer can shaped or pop can shaped, they land on the side so that the circular face is not the one that we see. But you have to be able to tell whether you're looking at it from the side or the top, or like some other angle, because if you're counting them, which I do, I have to identify them and count them for my work, for my research, and so I need to be able to identify them from all the different sides. And then also in a scanning electron microscope, which is what I present on Wednesday afternoons when I stream from my SEM lab, you can actually see all the different sides. So it looks like a three-dimensional object instead of, like, as I was mentioning, everything in the light microscope, we're just seeing through it. So we have to basically interpret what we're seeing. In the scanning electron microscope, the objects have, like, if it's the beer can, it looks like a beer can, like the way that we would see it. So you can actually see the circular part and the rectangular part, or the prismatic part. Are there tables or something where you can look it up? Not really. If you want help with diatom identification, though, my recommendation is Diatoms of North America, or this diatoms.org button here. It will help you. They often do give the girdle view, which is the view that we most commonly don't see. So there is a website that will help you if you're interested in learning diatom taxonomy, or for some reason you need to identify them. Thank you for the follow, Bison. In Arizona, we used diatomaceous earth for scorpion pest control. Yes, because the diatoms in diatomaceous earth are broken and fragmented, they can actually cut or get stuck on the hairs and legs of insects, and so they often deter them from moving through an area, and create problems and issues, so they can't path in a route that they would normally take. And we use diatomaceous earth that I bought from the hardware store on my sidewalk out front to get rid of ants occasionally, to try to keep them away from, like my daughter's always out there playing around. So to try to keep the ant colonies from expanding anymore, or whatever, we'll put a bunch of diatomaceous earth out there around them. People often use them in their gardens to get any kind of a pest, including slugs and snails. So anything like that that would create a problem for people, they'll use diatomaceous earth. Mirabox is the same thing for 1080p or 3045 for 4K. Yeah, so my camera has 4K output and the Camlink has 4K. Lets it move out 4K, but then OBS throttles everything at 1080. So, oh, you got the kitty going. I didn't notice that one. It must have popped up while I wasn't looking. Hopefully it worked for you. Thanks for using your channel points, Dangling. $20,000 for microscope. Yeah, it's an expensive one. It's only because of the differential interference contrast, and we use that mostly for imaging things for publication. So generally speaking, you don't need to have a fancy microscope like this unless you're looking at diatoms, in which case we kind of do. You can get by looking at diatoms in a cheaper microscope, but if you want to publish it, you probably can't. Yeah, so that's fine. Just let me know properly, and you can just have your husband message me or whatever, or post what he's trying to do, and we can try to help. What are the largest diatoms known, and what constrains them from evolving to be larger? That's a good question, Moth. The largest diatoms span upwards of 3 centimeters in size. You could effectively pick them up, but they're very long, skinny, and fragile, and they're not. They're shaped like some diatoms have like a stick shape, like this. We found some of these really, really long. Sorry, not 3 centimeters. 3 millimeters in size. Sorry, not centimeters. 3 millimeters. So like you could pick them up with tweezers, but you couldn't really like pick them up and move them around. They're marine diatoms, and they live in they're really long, like they would take up the whole field of view. In this field of view that we have, basically, they would take up most of it, and they're just very long, skinny diatoms. So they've managed to live in the ocean that way. So the question is, what constrains them from evolving to be larger? I guess a better question is, why would they want to be larger? So generally speaking, size is helpful for diatoms up to a point, but the larger you get, the problem is that it's harder for you to get enough energy to replicate quickly. An advantage of being larger is it's harder for things to eat you, but most of the diatoms are aiming to get large enough to be to avoid being eaten by certain predators, and the size of the predator's gape, or their mouth size, sort of controls whether or not they can be eaten. And we saw some of the predators, so the Cladocera, the Daphnia, the copepods, all those things that we saw, they're things that eat diatoms, and so they just have to get big enough that they don't fit in that organism's mouth, and then they're safe from being consumed by them. And it becomes energetically more expensive to make yourself bigger. Very small algae use almost exclusively diffusion to get their nutrients. They don't have to do anything other than just sit there, and the gradient in the water basically pushes nutrients into their bodies. So when you make yourself bigger, that becomes problematic, and the surface area to nutrient ratio and the gradient becomes much harder to manage, and they usually have to do something else. So I mean, most people know that diatoms are an algae, so they're getting their food or their energy in the form of sugars through photosynthesis, but it still requires that they have phosphorus and nitrogen and silica. And the larger you make yourself, the more of those nutrients you need. So I think there's sort of a balance for them with respect to size, how big they want to be. Another issue, besides just the sort of energetic side of it, is that making yourself larger makes necessarily almost always makes yourself heavier and denser, and so you sink faster. which is why the largest diatoms are usually found in the ocean, where the wave energy is a lot higher than it is in the lake system, for example. So it's easier for you to stay in the upper part of the water column when the wave energy is a lot higher, and there's upwelling currents and things like that that keep you afloat. So when you make yourself really big, you actually might make yourself too heavy, and then you'll have to sit on the bottom, and then you're out of the light, which is bad if you're plankton, but fine if you're a benthic diatom. So the largest ones are probably benthic diatoms that can lay around in the shallow water and can get enough nutrients to grow to that size. But that's an interesting question. I don't think anybody's ever asked me that one, so I like that. Thanks for the link. This is exactly what you're searching for. Okay, Cliff, no problem. Good luck. And if you have more questions, you can always drop in either the Discord or I stream, a couple times a week usually. You can feel free to ask. How long do they live was the question by Wabu, and the answer is, what do you mean by they? Because the problem with diatoms is the same problem that amoeba have, which is if I asked you how long does an amoeba live, there's almost no way to answer the question. And the reason for that is because amoeba are infinite organisms. What I mean by that is an amoeba, as you know, replicates by splitting itself in half. Like, now there's two of them. And if you're an organism, you used to be one organism and now you're two organisms. You can't tell which one is the parent or the daughter. They just are now two. And if you think about it and keep thinking about what's going to happen, it just keeps splitting and splitting and splitting and splitting. And so how do you put an age on any of them? If I asked you how old an amoeba lives, you'd be like, they're infinite. They literally live forever because they only split themselves. And so every one of them is basically genetically linked to the one that produced it. They don't have a cycle that we can think of like humans, where things can live and die. However, so to answer your question, diatoms have two stages of sexual reproduction. They have a asexual cycle like amoeba, where they can just split and split and split. But one of the problems is when diatoms split, they become smaller, or one of them stays the same size and one of them becomes a little bit smaller. And it has to do with the fact that they have a rigid external skeleton, and they have to make the parts for the next generation inside of the previous one. And so they necessarily have to make one valve that's a little bit smaller than the other one. And they form them inside their body because that's just the cells here. It's like a shape, like a pokeball. And I'm going to make it turn into two pokeballs. I've got to actually have a little one that fits inside of each of these. And then when they split apart, there's the smaller one from the original pokeball that now is a smaller cell than the parent cell. And then when that one splits, it also had one inside of it that's the smaller cell. And then it's a parent cell, and so it makes a smaller diatom, which makes a smaller diatom. So diatoms have a really interesting backwards growth cycle, which I usually try to tie to Benjamin Button, in the sense that the babies are huge and the ancient ones are tiny, and they get smaller as they get older instead of getting bigger as they get older. They don't grow to be big diatoms. They start off as babies, as big diatoms, and they get smaller as they reach adulthood. And when they get too small to fit enough stuff in anymore, they switch to sexual reproduction, and then they make a giant baby, and they start the cycle over again in a very backwards sort of approach. So they do have a sexual cycle, but the problem is that the sexual cycle is it's produced by the diatoms that turn into the males just busting open and gametes coming out, and the gametes go find another diatom that hasn't busted open, and it becomes an egg, and then that thing pops open and the giant baby egg forms, and it grows to a large sized baby, which then silicifies and now I have a giant baby. But I mean, it's hard to tell where's the start and the end of their life cycle, because if you count the baby as the origin, and the parents as the end of that cycle after many generations, it's rough on our concept of age. But I will answer that question in a more straightforward fashion. If I have a living cell, a clone, it has a lifespan as well. And their lifespans can range from a couple of days to a month, for each one of the clones. And then they can replicate many times before they reach adulthood. So each one of those clones has a few days or a month or something that they'll be alive cells. And then they can actually insist, which is another problem for our model of like age, because they can basically do like a hibernation phase, and when they do that, then they just basically clamp down and shut themselves off, and just persist, and they can persist over an entire winter and then come right back into a living state when the season starts again. And so, like, I mean, we have a very narrow concept of life and birth and death, and it doesn't work very well in the microscopic world. But to answer your question, an individual stormtrooper in this scenario lives maybe a month, or some of them might live a couple of months if they're benthic organisms, and then the, the clone is probably part of a living organism that has a much longer lifespan. And the lifespan of the organism, in the sense of how long before sexual cycles happen, can be months to years. Some observations of the sexual reproduction cycle in diatoms suggest that they can be 3 to 5 years. So the diatoms can live 3 or 5 years before they reach the sexual reproduction point again. So it's a really challenging a problem to answer. Diatoms are very weird, yes, moths. Thank you for the compliment, I guess. You're about to go play Skyrim with some family. It was nice chilling with you and chatting. Yeah, thanks for hanging out, Xanophyte. So hopefully that makes sense, Wabu. I'm sorry I'm blowing your mind, Moss per hour. There's a lot to think about, and I've thought about most of it. So I'm going to look at one more sample. I don't know how long. How long have I been streaming? 2 hours and 21 minutes. I think I'll just do one more sample. And then, Kalathon, thank you for the subscription. Always 19 months. It's like from the very beginning when I first started streaming, Kalathon's been here subscribing. Just like forever. The family tree is it's a circle. It pretty much is. You're old and I'm old, and we can be old together. It's perfect. Thank you for your subscription and for constantly supporting our channel. You know that all of the money that comes in goes to my students' research and resupplying stuff for the lab. I don't take any money home, and so it's important to me when people do donate money through whatever process that they do, that I recognize that that is a contribution that goes directly to my students and helps them be successful. So 19 months times $2.50 is a chunk of change that you've donated and that you can feel happy about having contributed directly to student research. So that's cool. I don't know if this is actually going to produce anything. This like that sample did not look like it had very much stuff in it. It was like all in a clump and it didn't want to separate. So I'm going to try it, and then I'm going to stick on, if this doesn't have anything in it, then we'll look at the Lemna sample. Oh, it's got some stuff in it, but it's just some loose diatoms. Oh, here's something. What is this? A euglena. So I haven't seen a euglena before today. I mean, just today I've seen one before. There's a bunch of euglena. There's another one. Let's zoom in and look at it at the 20x since I've got a cover slip on it. There's a euglena. Euglenoid. I guess I should be more careful. It's a pretty little one. Usually they have this sort of teardrop shape. Euglena are like some of the other organisms that we talked about. They can... what's going on? I need to make some adjustment, but I can't figure out what. Yeah, that's good. They can switch between being autotrophs and being heterotrophs, depending on what is more available in their habitat. So this one has chloroplasts. They usually have chloroplasts, but if they're in darkness, they'll switch to eating organic matter or eating other organisms. It's kind of cool. Can diatoms hybridize? Is that a thing? Do you mean like make mule, like offspring that are created from sexual reproduction of two different species? I don't think so, but I don't know the answer to that, Moss. I don't actually grow diatoms and watch what happens with them, but I think that they can only mate with their own species, but I don't know for sure. It's really pretty. These little things have somewhere in their body there's a flagella at the end of that tail, usually. Sometimes it's not obvious or it's really tiny, but they have this sort of typical shape, and then chloroplasts in their body, and usually you can find a little red eye for them as well, somewhere in there. That's a clue. There's also some, looks like Oligosyra here. There's some sort of diatom. There's some more algae. I have a lot in here though. It's pretty sparse. There's some sort of an egg. There's a diatom. There's lots of diatoms in here, not much else. Well, it'll be okay. We can look at the sample and we'll digest it a little bit with acids and see what we can find. It looks like it's mostly just has some euglena in it. There was a piece of plastic that I just passed, and a bunch of naviculoid diatoms. Oops, here's a swimmer. That is a rotifer. It's a very fast-moving rotifer. Piper Lee, are you still here? I don't know if you gave up on it yet. If you are, and your husband is going to stream stuff from Microscope, I'd be happy to start shouting him out. I have a little thing that rotates that says something like up there above where it's announcing Freckled Science. Yeah, you're still here. So if he has a Twitch account and he's going to stream into Twitch, I don't know what he does with his Microscope, but if he's going to do that, let me know, and I'll add him to my little list of Microscope streamers. So once he gets it sorted out, of course. Okay, that's simple, super boring. Let's look at the Lemna one, and then we'll call it a day for looking through these samples. That was the swamp scum. This is the swamp Lemna. Lemna is just another word for duckweed, so not sure if he's going to stream, but he definitely wants to be able to take pictures. Oh, okay. Well, if he just wants to take pictures, he can mount a camera on there without having to mount the streaming stuff on here. That's a lot simpler as long as you have a phototube. If his Microscope doesn't have a phototube, there's usually a way that you can mount a camera onto the eyepiece as a way to take pictures. And also, if he doesn't have a fancy camera like I do, but you want to take pictures in your Microscope, they make these little simple iPhone mounts. So the problem with the iPhone trying to take a picture into a Microscope is that you have to have it at like a perfect angle to get the Microscope circle basically out of your way. they make little things that you can even 3D print some of them that will hold an iPhone or a camera, a smartphone onto a microscope, and then you can just take pictures with your phone. They're not bad in terms of like getting decent pictures. So it's an option. He's an engineer, so he knows about materials, but likes looking at pond water and stuff. Yeah, it's fine. So if you're trying to find software for the camera, okay. So yeah, that might be a separate question, but you can just bring it into OBS. So you already know how to use OBS, any camera, through the cam link, and then see how it goes. But yeah, let me know and I'll see if I can try to figure something out. That would be great. We like helping people. I like helping people learn how to do stuff with microscopes. So what's in here? I don't know what that little thing is. Oftentimes when I'm looking around at samples, I don't know what I'm looking at. Speaking of which, I don't know what this is. And it's just taken me a while of like using guidebooks and things to try to figure out what I'm looking at. If it's a diatom, I know those pretty well. If it's something else, I don't know. I don't know what this is, for example. I have no idea what we're looking at. It's a ciliate, and that's the limit of what I know. It is some kind of a ciliate. I've never seen anything shaped like this, and I don't know what's going on right now with it. It's some kind of a ciliate. So, protozoan. But that's as far as I could figure out. I don't know if there's more of them, but that shape is pretty unique. So I will see if I can figure out what it is. There's a diatom. I'm seeing a bunch of these little things, which I don't know what they are. Like a donut. There's a diatom. All the diatoms look like little boats in here. This was taken from the bottom of some duckweed. Duckweed are actually kind of interesting. Most people interact with duckweed as it grows over their pond, and then they're very angry about it. But lemna, the true duckweed, is a type of floating macrophyte. They have what's called a pendulous root. In other words, that like plants usually have a root. The lemna have a root that just hangs in the water, and like some pistia and some of the other macrophytes in lake systems, they just have a pendulous root that hangs from the plant, and so they get all their nutrients from the water directly. They don't have a root that goes into sediments, and so you can just pick them up and pull them out of the water or skim them off of the surface. And there's whole classifications of macrophytes or plants that are found in aquatic systems that have this sort of characteristic of floating leaf macrophytes, where they're just basically hanging out in the water and could be pushed around by anything, and they don't have any kind of root in the water itself. So I mean, just a hanging root. They don't have any roots in the sediments. So this is something that I teach my students in the Lakes and Wetlands class. We look at different types of macrophytes. We look at different types of aquatic insects. We look at different types of worms. I try to make them learn a little bit about the zooplankton, the phytoplankton. I force them to learn some of the differences between the different types of algae. We learn about the physical constructs of the lake, like thermoclines and oh, I see what's going on. I got some water on my objective. I was trying to figure out what this little guy was. That's an ostracod. Come back, little ostracod. Yeah, that's an ostracod. So not a Cladocera. Its entire body is inside the shell. There's no head separate from the rest of the body, like all the little things that we had seen. It went outside the cover slip. That's why the background changed colors. It doesn't care that there's a cover slip. And it has a calcareous skeleton that's a little bit heavier than the Cladoceras skeletons that we saw just outside the cover slip again. My Etch-a-Sketch skills are being tested. So I don't know. Ostracods are kind of cool. They look like little jelly beans. They fossilize in the lake records. I force them to learn, yes, and not teach them. I feel like that's the way they interact with me a lot of times. Like, why are you making me learn stuff? Also, just as a technicality, Indiana does not think that professors are educators, so I like to intentionally say things like that. I force them to learn things. Yeah, so we got some weird things in here. I think we did a good job of sort of touching on all of the samples that I collected. I was pretty excited to be able to look through everything. This is a light microscope stream, so if this is your first time observing what we're doing here, I would say most frequently I stream from the scanning electron microscope. Sometimes I stream from my light microscope in the evenings when I'm home. I'll probably stream a little bit more frequently from the SEM in the coming time, but it's usually like 2 to 5 or something like that on Wednesday, and then like I go home from work, so it's usually not after 5. And then I sometimes do streams of, like, last night was it last night? Yeah, last night we did the moon. I sometimes will just do like photography streams where I put the moon or birds, or I did snowflakes and macro photography streams. So I'm kind of all over the place, but it's usually trying to take something very little and trying to make it big, whether it's something that's actually very big up in the sky that looks little, or something that's on planet Earth that's very small and I want to make it look big. But that's what most of my streams are. And sometimes I do art streams where I colorize the SEM images or edit my photos. So if you want to get a sense of what I do and you haven't been here before, that's sort of it. That's what I do. And then I like to talk science. So it's an interesting technicality of Indiana. Yeah. Well, I will tell you, when it was like educators are allowed to get their COVID vaccinations, they did not include the professors in that list. Oh, I forgot woodworking. I do sometimes do woodworking streams. And those are sort of hobby things for me, photography and woodworking. So I do sometimes build stuff. Musical instruments, cutting boards, furniture, whatever. I have to build my daughter's room is being turned into some sort of a forest landscape with some sort of really fancy forest wallpaper with foxes and stuff hidden in it, because that's her favorite. And she has like a little sort of bay window. There goes that ostracod. And my wife wants me to put in a sort of bench / shelf thing, so I'll probably have to do that. I don't know if I'll stream it, but that'll probably be the next thing I build. And then but whatever, I try to do that to get away from I like to do woodworking and stuff to get away from thinking about work, photography, woodworking, whatever. Okay, we've been streaming for a while, and I feel like I've had enough of it, and you probably have all had enough of it. So we'll raid out to somebody. I want to find someone for me to raid to, but I'm going to start that, and then I'll come back and chat just a little bit. Why don't we go over to Volcano Doc, since how we're doing geology-like stream, Earth stuff. Volcano Doc is one of my friends, so she's a scientist. She has a PhD and she studies volcanoes, and she just talks about volcano stuff all the time. Volcanoes are cool, so we'll do that. What kind of instruments do I build? I'm working on building a tongue drum right now. I've got a prototype that I made out of cedar, but I have some really nice wood I want to try to make one out of. I also make musical shakers, like little wooden shaking instruments. So if you're a musician and you stream and you want a little wooden shaker, just let me know. I can make some. I did that for Asymmetrical Soul. I made a kalimba for Hannah Rebecca Music. So these are friends of mine here that stream. This is Asymmetrical Soul. So he has a shaker that I built. You can open it up and actually change out the shaking components to something else, but it's made out of hardwood. And then my friend Hannah, who wrote a diatom song for my channel, this one, is pretty incredible. And because she wrote me a song, I made her a kalimba, but she didn't know. And so I built it on stream, and then I showed up and showed it to her while she was streaming. So pretty cool. All right, let's go visit with Volcano Doc. Thanks everybody for hanging out. Thanks for the resubscription from Kalathon and all the follows.